In this lesson today, I'll talk about the replication of DNA. As we have already discussed in mitotic cell cycle, that DNA has to be duplicated before a cell divides. For instance, if there is a human cell which has 46 chromosomes in it, and it divides into two daughter cells by mitosis, each having 46 chromosomes in it. So you see, from one parent cell which has 46 chromosomes, it is divided into two cells, each having 46 chromosomes. So this additional 46 chromosomes comes from the duplication of DNA, which is known as replication during S phase in an interphase of a cell. In this lesson, we'll discuss the method of semi-conservative replication and the role of different enzymes in replication. We will also discuss the other theories that describe the possible methods of DNA replication. However, the Mendelssohn's and Stahl's experiments prove that DNA replicates only by semi-conservative replication, not by any other process. However, in this lesson, we will not discuss Mendelssohn's and Stahl's experiment. We'll discuss that in another video. So let's see replication. Replication means synthesis of two DNA molecules from a single DNA molecule. If this is, for instance, a single DNA molecule, there are two DNA molecules synthesized from this. Single DNA molecule. This process is known as replication. Both these DNA molecules are two identical DNA molecules, and they are synthesized from or made from single DNA molecule. So this process is known as replication or maybe duplication of DNA. There have been three theories proposed to explain the process of replication. The first one is semi-conservative replication of DNA. Semi-conservative replication of DNA. The other is conservative replication of DNA. And the third is dispersive replication of DNA. Semi-conservative replication describes the formation of two DNA molecules from one DNA molecule in such a way that each newly synthesized DNA molecule has one old strand and one new strand formed with it. So half of the DNA is conserved in each of the newly synthesized DNA molecules. So that is why it is called semi-conservative replication. In conservative replication, however, both the old strands of the DNA molecule are conserved and a new DNA molecule is synthesized with both the new strands of DNA. So whole of the DNA molecule is conserved in unchanged in this method. So this was named as or suggested as conservative replication of DNA. In dispersive replication, it was suggested that each newly synthesized DNA molecule has fragments of old and new DNA molecule present in it. It was later proven that DNA does not replicate by conservative and dispersive methods. It was proven that DNA replicates by semi-conservative replication. So let's see semi-conservative replication and the role of enzymes in semi-conservative replication. For instance, if this is the DNA molecule, that has to be synthesized or that has to undergo duplication and synthesize two identical DNA molecules. It may be coiled and condensed, so an enzyme attaches with it known as a gyrase enzyme, DNA gyrase, and another enzyme known as DNA helicase attaches with it, and it dissociates hydrogen bonds in between the two strands of DNA and separate the two strands apart from each other. If this has to be magnified and viewed, imagined, this has, the two strands have been separated apart by breaking down the hydrogen bonds in between the specific base pairs by this helicase enzyme. This is helicase enzyme. We already know that these two strands are anti-parallel to each other, and they have their specific ends that are represented as, if it is 5' on this end, the other strand will have 3' end on this side, and so on the other end this has 3', and the other strand will have 5' here. Each DNA molecule has the specific base pairing. So if this upper DNA molecule, upper DNA strand has an adenine base here, the lower will have thymine here. If this has guanine here, this will have cytosine here. If this is cytosine here, this will have guanine here. So this is specific base pairing that you already know. We have discussed in the structure of DNA. So you can write any sequence of bases on one of the strand, and the other strand will have just the complementary bases on it. So there is no specific order to write. DNA would obviously have the specific order of these nucleotides on each strand, and the other strand would have complementary to that. But this point, you do not need to learn some specific order of these bases on a DNA molecule. So I'm just writing it randomly, but complementary on both of the strands. Okay. An enzyme known as DNA primase attaches with this one strand. So this is DNA primase. DNA primase attaches a short sequence of nucleotides, which is complementary to, obviously, this strand, and this is known as an RNA primer. So this RNA primer will have 5' end on this side and 3' end on this side. After this primer has been attached with this DNA single strand, another enzyme, DNA polymerase, attaches with this single strand of DNA, and attaches free nucleotides with this chain of primer on 3 end. So it attaches free nucleotides here. This has adenine attached here. This has thymine attached here. This has cytosine attached here. So this chain continues to grow in this direction. The free nucleotides are attached from 5 to 3 end, and this chain continues to grow in this direction. Helicase continues to unzip this DNA strand, and the DNA polymerase moves in the same direction and keeps on growing this chain from 5 to 3 direction. So that is why this is known as a continuous strand, or maybe known as leading strand. This is known as leading strand, or it is known as continuous strand. However, on the other end, the newly synthesized DNA strand is formed in fragments, as the newly synthesized chain will always grow in, or will add new nucleotides in 5 to 3 end, and it would be complementary to the template strand. So the newly synthesized DNA strand on the lower strand will be in an opposite direction. So it will have 5' on this side and 3' on this side. Helicase will keep on moving and unwinding and unzipping these two strands apart. So every time the DNA polymerase, that would make a new fragment from 5 to 3 direction. So this would make a discontinuous chain in fragments. These fragments are known as Okazaki fragments, and this is also known as lagging strand. Now these fragments have to be joined together. Another enzyme called ligase enzyme joins these two strands together by a phosphodiester linkage in between them, and that makes a continuous strand by joining these fragments together. So this is DNA ligase. So we have discussed gyrase enzyme, which uncoils the DNA molecule, DNA helicase, which breaks hydrogen bonds and separates the two strands of a DNA molecule apart, making a replicating fork. Then we discussed the role of DNA primase, which attaches a primer. This primer is essential to be attached with the single-stranded DNA, as this DNA polymerase cannot initiate the joining of the nucleotides with this single-stranded DNA. However, this DNA polymerase that attaches the new nucleotides, the free nucleotides, with the 3 end of the primer and grows this chain from 5 to 3 end. Another enzyme we discussed is DNA polymerase number 4 and DNA ligase. The difference in between the DNA polymerase and the DNA ligase is the DNA polymerase attaches single free nucleotides with the growing chain of the DNA strand or the nucleotide chain, whereas the DNA ligase that joins the two fragments of the polynucleotide chains together, not the free nucleotides.